Hello and welcome. Thank you for joining us for the second annual Black and Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we would like to thank our major sponsors, Immune Deficiency Foundation and the Imperial College London. Don't forget to tweet today using hashtag #BII@thebench, with each word capitalized for accessibility purposes. We will also have live captions and ASL interpreters during the session. You can submit questions through the comment function on the platform, not the Q&A. And as a reminder, we will require all audience members to be polite and respectful while in the Black and Immuno space. If we see anything offensive, your comment will be deleted immediately and you will be banned from the platform. At the conclusion of the week, recorded videos will be posted on our YouTube channel and transcripts will be made available on our website, blackandimmuno.org. My name is Heather Caslin, and I'm a fourth-year postdoc at Vanderbilt University and a volunteer for Black and Immuno. I will be the moderator for today's basic immunology session. We have 4 great speakers here to tell us about their work. They have about 20 minutes for their presentations, and then we will follow with a 5-minute Q&A. Without further ado, let's jump on in. Our first speaker for this session is Dr. Yasin Elfaki. Dr. Alfaki received his BSc in Medical Laboratory Science and Microbiology in 2009 from Sudan University of Science and Technology in Khartoum, Sudan. He received his MSc in Medical Laboratory Science, Microbiology in 2013 while working at the same university as a teaching assistant and then as a lecturer. In 2015, he was awarded a scholarship from the German Academic Exchange Service to do his PhD at the Helmholtz Center for Infection Research in Braunschweig, Germany. Under the supervision of Professor Jochen Huhn, Dr. Elfaki's work focused on regulatory T-cell development during influenza A infection. Since 2019, after obtaining his PhD, he's been working as a postdoctoral research fellow at the University College London Institute of Immunity and Transplantation in London, UK. His work in the lab of Professor Luke C. Walker has been on innate lymphoid cells in type 1 diabetes. Welcome, Dr. Elfaki.
Thank you very much. Thank you for having me. Okay, if I may share my screen. Let me know if you can see it. Let me know if you can see it and see the laser pointer. Okay. Thank you very much for the introduction, and thank you very much for the invitation to speak at Black and Immuno. So I'm going to talk about my work, which is involving addressing innate lymphoid cells during type 1 diabetes. So type 1 diabetes is a disease, is a public health concern. This disease is estimated to affect around 400,000 people in the UK, and almost 30,000 of them are children. The disease is growing by an alarming 4% each year, the incidence of the disease, and by 5% among children under 5. And it's estimated that a person with type 1 diabetes will have around 65,000 injections and measure their blood glucose more than 80,000 times a year and in their lifetime. And this actually indicates and denotes the intensity of management that this disease requires. So the pathogenesis of this disease starts with the damage of the islets and specifically of the beta cells of pancreas that produce insulin. And insulin is a hormone that regulates blood sugar. And when you have damage of the islets by immune cells, so this is why it's an autoimmune destruction. So when you have damage of these cells by principally T cells, but we also know that other cells can participate in that, you have eventually a deficiency in insulin and thereby hyperglycemia and all of the consequences and complications associated with that. So we know that in mouse as well as in humans, beta cells can be attacked by T cells and B cells. So these cells participate in the autoimmune attack on beta cells. However, little is known actually about the participation of other cells and whether other immune cells are involved in this attack. So although we are a lab that has a long-standing tradition in addressing T cell responses in type 1 diabetes, and specifically Tregs, we've found this cell population while we were looking at regulatory T cells. So we were looking at CD4 positive, CD25 positive proportion of cells in pancreas of diabetic mice. And we found that there is a sizable population of cells that lack the expression of CD4 while expressing CD25. This population was absent in pancreatic as well as peripheral lymph nodes. So we went on and stained for the typical lineage markers in order to define this population. So we stained for CD3, CD9, so for lineage markers that indicate or mark T cells, B lymphocytes, dendritic cells, NK cells, macrophages, and other myeloid cells. And what we've seen is that this population lacked the expression of these typical lineage markers. So this led us to think about innate lymphoid cells. So what are innate lymphoid cells? So innate lymphoid cells are innate cells. So that means they lack the expression of antigen-specific receptors. They are deeply tissue intertwined. So they are found within tissues and they perform multiple functions. But one of the most important functions of these cells, so immunological function, is that they act as a rapid source of copious amounts of signature cytokines that otherwise are elaborated by the adaptive immune cells, by helper T cells and the different subsets of helper T cells. So for instance, if you have a tumor or if you have an intracellular infection like a virus infection, then ILC1s will act very rapidly and produce huge amounts of the signature cytokine interferon gamma, which acts by activating macrophages as well as encouraging cytotoxicity. Group 2 innate lymphoid cells, on the other hand, they act when you have a large extracellular parasitic infection, or in their case of allergy, they produce the typical type 2 cytokines. And one of these typical type 2 cytokines is Amphiregulin, which is a growth factor that actually is important in maintenance of tissue integrity following inflammation. We also have ILC3s, which act similarly to Th17 cells, in that they elaborate IL-17 when there is an extracellular microbial infection. So we wanted to see whether our cells are typical of ILC description. So first, they lack the lineage markers, but also they express CD127, which is a pan-ILC marker. Not only that, but also these cells, the CD25 positive, CD4 negative cells that we found in the pancreas, they express typical markers that are associated with ILC2s. So I'm talking about T1ST2, which is a subunit of IL-33 receptor. We also have other markers like KLRG1 and also inducible co-stimulator ICOS, as well as others. So this led us to think that the cell types that we are after now are group 2 innate lymphoid cells. To be absolutely sure of that, we stained for transcription factors, which are known to mark the ILC populations. And what we can see here is that our population, the CD25 positive population, specifically expresses GATA3, but lacks the expression of ROR gamma T and TBET, which is typical of ILC2s. And that means that our cell type that we're looking at here is typical of ILC2s or group 2 innate lymphoid cells. Not only that, but also we checked for cytokine expression. And this cell population also was producing IL-5, IL-13, but these cells lacked IL-17 and interferon gamma, suggesting and further indicating that these cells are actually ILC2s. So we also checked in order to make sure if those cell types are can be found in mice that lack the adaptive immune compartment. So RAG knockout mice, these mice, they lack the machinery that is involved or that is required to generate antigen-specific receptors, and thereby they don't have T or B cells. And we could find the CD25 positive, CD127 positive ILC2 population in these mice. However, when we also check the pancreas of NSG mice, which lack signaling through the common gamma chain, and that means they don't signal through important cytokines for lymphocyte development, so IL-7 and IL-2, and consistently, those mice, they do not have this cell type. They do not have cell type, and this is actually confirming or suggesting that these are ILCs. So having shown that we can find these cells in the pancreas of mice, the question is, do they respond to T cell infiltration during type 1 diabetes? In order to study this, or in order to address this question, we've made use of this following mouse, a mouse that expresses two transgenes. The first transgene is a model antigen that is expressed under the rat insulin promoter. That means this mimics an expression of tissue-specific self-protein. That means it's expressed only in the pancreas. The other transgene is a TCR, so a T-cell receptor transgene that specifically recognizes this OVA albumin. So it's a DO11 TCR. And this mimics then the expression or the generation of self-reactive T cells. So in these double transgenic mice, what you see is an autoimmune attack, so an infiltration of autoimmune cells or immune cells in the islets of Langerhans. And if you measure blood glucose of these mice, you see an elevation of blood glucose over time, so indicating that these mice develop diabetes, so they develop hyperglycemia and diabetes. So in order to address ILC2 in this regard, we've devised the following gating strategy. So the gating strategy consists of first looking in pancreas of murine pancreas. We first look at CD45 positive hematopoietic cells and then gate on lymphocytes by size and then gate out doublets. We have a cocktail of lineage markers. So we stain for CD3, CD19, CD11B, CD11C, CD5, as well as others. And to get rid of all the cells that might inadvertently contaminate our CD127 gate, and as I've just indicated, CD127 is used as a pan-ILC marker. So gating on CD127 plus lineage negative, we then can characterize ILCs, and as you may appreciate here, most of these, or the vast majority of these, are GATA3 high, which is indicative of their ILC2 phenotype. So now we want to see whether they are affected. So first we checked their proliferation, and for that we stained in both double transgenic mice, so the diabetic mice, so the mice that have both transgenes, and compared them with their single transgene controls, littermate controls. We stained for Ki67, which is a transcription factor expressed by proliferating cells, and at steady state, ILC2s, which is known in the literature, they don't really proliferate that much, but you can see that in diabetic mice, proliferation is significantly increased. We have a positive control T cells, which we know they proliferate in response in diabetic mice because they are the cells that derive the disease. However, not all innate immune cells that we looked at have the same response, because NK cells, for instance, they don't increase their proliferation in response to diabetes. So this showed us that ILC2s actually respond by increasing their proliferation, but what about their effector function? So this is why in the gated ILC2s, we checked for effector cytokine production. And we looked for IL-13, and one thing that was apparent to us is that ILC2s increase their IL-13 production, as evidenced by actually the increase in proportion of IL-13 positive cells among ILC2s. Not only that, but also the amount of IL-13 produced on a per cell basis is significantly elevated. So as indicated here by the measurement of mean fluorescence intensity. Not only that, but also we checked Amphiregulin, which I talked about is an important growth factor that is essential for maintenance of tissue integrity following inflammation and limiting of the immunopathology following inflammatory stimuli. We see that there is a significant increase in the frequency of Amphiregulin positive ILC2s, and also a significant increase on the amount of Amphiregulin produced on a per cell basis by ILC2s in diabetic mice compared to the litter-mate controls. So these data indicate that ILC2s do not only respond by increasing their proliferation, but they also respond by increasing the production of their effector cytokines. So I'm at the end, actually, and I want to sum up what I told you now. So type 1 diabetes is a public health concern, and there is a growing interest in understanding how different cell types are responding and contributing to this disease in order to manipulate them. And this is the premise of immunotherapy for type 1 diabetes. So in both in mouse as well as in human, although I haven't shown you this because of time, but we know that innate lymphoid cells, they reside in the pancreas and they're present there, as well as adaptive immune cells. They don't only reside there, but they also respond to diabetes induced by T-cell infiltration in the mouse model that I just described. And this is actually by increasing their proliferation, but also increasing the production of their effector cytokines. I've shown you examples of IL-13 and Amphiregulin. And now what we're doing actually is trying to understand the functional relevance of ILC2 T-cell interplay. And in order to do that, for instance, we've generated RNA sequencing data of ILC2s from diabetic as well as control mice to understand what is the influence of T-cell infiltration of ILC2s and whether we can pin down any molecular pathways that are initiated on ILC2s by infiltration of T cells. And also one thing that we want to do next is to address the big question is that whether ILC2s are good or bad for type 1 diabetes development, whether they're helping the initiating cells or whether they are otherwise limiting the inflammation, which is not an easy question, I have to say, to address. So with that, I'm at the end, and I want to thank all the people who are involved in the study. So the Walker Lab, Lucy Walker, for giving me this exciting talk, this exciting project, members of the Walker Lab, past and present, the funding, as well as collaborators. And also Black in Immuno, of course, for giving me this exciting opportunity to speak and to give you an idea about what I'm working on. If you're interested in hearing more about my journey as a Black immunologist in academia, I was hosted by the Abraham Institute last month, and the talk, and I was talking exactly about that, and the talk is now available on YouTube. So if you go to the Abraham Institute YouTube channel, you can find the talk and you can watch it. With that, I'm at the end, and I'm happy to take questions. Thank you.
Thank you, Dr. Elfaki, for such a great talk on how ILC2s are resident in the pancreas and how they increase with type 1 diabetes. I'm looking for questions in the comments if anyone has one, but I can start off. Has anyone used IL-13 or Amphiregulin to try to treat or prevent type 1 diabetes before?
Not to my knowledge. So I haven't seen anything that would pinpoint to this. Mainly, I think because at least for ILC2s, there hasn't been a role ascribed to ILC2s in type 1 diabetes. This is what we're trying to do, actually. But haven't seen something like this.
I haven't either. We do more like type 2 diabetes type work, but I like to kind of keep up with the literature in both. This is a question. I don't know a lot about ILC2s. Are there models you can use that can increase ILC2s either just generally in the body or in a specific organ to try to test like if they're helpful or hurtful?
Yes. So the model that people use is the injection of IL-33. So
Okay.
IL-33 is a conserved inducer, let's say, of ILC2s. So whenever you inject it, they can even appear in organs that otherwise they wouldn't be present to, like in the liver, for instance. If you inject the mice with IL-33, you can find ILC2s everywhere. So yeah, I think that's probably, I have to say other genetic models are very lacking because, of course, ILC2s share a lot of, they're not specific markers in order to pinpoint ILC2s, to overexpress them or to dampen them with it. So it's a bit tricky.
One of the other audience members, asked, is there a way to delete them genetically?
So basically, genetically, there are some ways, but they are tough. So for instance, you can use a mouse that was described by Andrew McKenzie in Cambridge. They used a specific deletion of ICOS in ILC2s. We can do that, but I have to say here, we didn't see an increase. I mean, we see an increase in ICOS, but I don't know how specific that is for ILC2s in our systems. So this is one way to do it. But people use also GATA3, but the problem is with GATA3 is that you can also delete Th2 cells, you can delete Tregs that are co-expressing GATA3, you can even, because GATA3 is expressed virtually everywhere. So it's a bit tough.
See, two more questions. Do you think that the ILC2 proliferation and activity is due to breached tolerance? Are they generally suppressing the T cell response, or can you hypothesize on that?
So the thing is, we force, of course, here breach tolerance. So we know that this is in response to T cell breaking away from their tolerant behavior, because, of course, we force the expression of self-antigen, and we force the expression of a TCR, self-reactive TCR. So we think that this is because of T cell infiltration. Whether they are good or bad, so that's a bit tough. We don't have any answers yet. If I would bet, of course, I would say, okay, maybe ILC2s are good, because in other situations, Amphiregulin production, for instance, you could hypothesize that it's good, because in influenza infection, for instance, if you delete Amphiregulin on ILC2s, the mice die not because of increased viral load, but because of the increased immunopathology. And there is no tissue repair because there is no Amphiregulin. So you would assume something like this here, but I can only speculate.
Kind of a related question. Have you run a time course experiment to check the dynamics? Is it happening like right at the same time that you see T cell infiltration?
I have done this. So I have looked at them when they were with the mice are young, before development of diabetes, when the mice are developing diabetes, and also at a late time point. And it seems like it goes hand in hand, because actually also T cell infiltration happens before diabetes development, because there has to be encephalitis and inflammation of the islets before diabetes actually fully ensues. And at that time point, you also see a response, but we don't see a proliferative response, but we see shift in balance of ILC2 to ILC1. It's something that I haven't alluded to in my talk, but this is something that we see. So we see responses, and it seems like it goes hand in hand with T cell infiltration.
We have a few questions more in the chat. So as we're done, if you want to jump in the chat and answer, but I'll have one last question. This is from one of our Black and Immuno co-founders. We know that ILCs are important in the interaction of the microbiome with the immune system. Do you know if there's a microbiome type 1 diabetes interaction?
That's a tough question. I don't know. We would have to do the experiments, of course. We would have to house our mice under germ-free conditions and then see whether their phenotype is influenced by type 1 diabetes, or their phenotype is different in type 1 diabetes. Of course, we would need still T cell to do that or to initiate the disease. So I don't know. I don't know actually if ILC2s are, they require microbiome or not for their development or their function. I have no idea.
I don't either. Well, thank you so much for that great talk.
Thank you. Thank you.
Next, we are going to have Dr. Ukpong Eyo. Dr. Eyo was born in Nigeria and grew up in several different countries. He immigrated to the U.S. in 2003 to pursue undergraduate studies at Northwestern Missouri State University. He then went on to graduate school at the University of Iowa, where he developed a keen interest in real-time imaging of microglia during development under the mentorship of Dr. Michael Daly. Following his PhD studies, Eyo joined the lab of Dr. Long Jun Woo at Rutgers University in New Jersey and then at Mayo Clinic in Minnesota to study microglial neuronal connections. In August 2018, Eyo started his independent lab at the Department of Neuroscience in the Center for Brain, Immunology, and Glia to continue his research on microglia with a focus on the developing brain. Eyo has a long-standing interest in microglia, myeloid cells that represent the major immune cell lineage in the resting brain parenchyma. In the 3 years Eyo has been independent, he has been immensely successful with several short-term and 2 new R01 awards supporting his work, and 13 publications. That's so great. Eyo is an engaging speaker, and his insights into the behavior of a poorly understood immune cell type in the immunologically distinct anatomical compartment should be of great interest to all of us in the audience today. His career path and success also make him an important role model for graduate students and postdocs as they pursue their own developing careers. Away from the lab, Eyo enjoys his time with his blessed family, including his wife, 2 sons, and 2 daughters. Welcome, Dr. Eyo.
Thank you so much. I haven't used this platform really much. I just want, can everybody see this? Can you see my screen?
Yes, we can.
All right, let me, is this in full screen mode now? Or is this in the, do I have to swap the display?
Swap the display.
All right. So are we all good now?
Yes.
All right, so it's such a delight to be here and to show you some of our work and our talk. Thank you so much for the introduction. Thank you for the invitation to give this talk. So I'm a little different. I study actually the nervous system, but I study the immune cells or the primary immune cell in the nervous system, the myeloid cell, the microglia that are resident in the brain. And I'm hoping that I can learn some from traditional or maybe more realistic immunologists than myself, who's more focused on the brain. In any case, these cells are the microglia. They're actually unique in respect to all the different cell types of the brain, because unlike other cell types in the brain, they actually born outside the brain and then migrate into the brain later on and take up residence there. What is fascinating is that we now know that the brain is actually quite dimorphic or sexually biased, where female brains often behave differently than male brains. And this is actually shown in the course of different brain disorders. So this is a schematic over here showing some disorders that are generally female biased. So you have anxiety, depression. In the case of multiple sclerosis, it seems to predominantly affect women over men. But then there are other conditions that are actually predominantly male biased, the famous of which, of course, is autism, but also ADHD, attention deficit disorders are also more common in males over females. And we wonder if it's possible that these microglia could be playing a role in regulating some of the behaviors that are present in these disorders. So the approach that I'm taking here is we look at cells, and in this case, the cells we're interested in is the microglia, and potentially that microglial cell may regulate the circuits in the brain in the form of neuronal interactions that regulate these behaviors. And so presumably the pathway that we're looking at is the potential ability for these cells to be perturbed, and then by their perturbation affect the circuits that then regulate the behaviors. And what I'll be showing you is data that we have. Again, this is unpublished work. This is actually work that's being done by 2 students in the lab, mostly a graduate student and a postdoc in the lab. But we took advantage of the fact that some studies had been done several years ago looking at microglia. And although they are macrophages, so they are myeloid cells and similar to other macrophages outside the brain, they're actually quite distinct, both from other cell types in the brain, as well as from macrophages outside the brain. And this is one of the early RNA-seq studies that was done by Al-Khoury's group, where they showed that several transcripts in microglia, shown here in black, are highly expressed in microglia and largely absent in the brain. But similarly, those same transcripts are often highly expressed by microglia and not present in peripheral macrophages, or in this case, peritoneal macrophages. And one of the most highly expressed genes in this case is this P2RY12 gene. This gene, again, is uniquely expressed by microglia in the brain. It's not expressed by other cell types in the brain, but it's also distinguishes microglia from other peripheral cells. So my work in the lab has started to focus on these receptor and this gene and what it potentially can be doing. This is previous work, again, from several years ago that actually took macrophages, or actually hematopoietic stem cells, and put them in the brain to ask the question whether it's the brain environment that confers this unique identity on the microglia. And when they took out microglia from the brain, either in adults or from the fetus or from the yolk sac that eventually gives rise to microglia and other macrophages, they could see that when you put those hematopoietic cells back in the brain, they recapitulate the microglial phenotype here in yellow. However, when you take differentiated liver macrophages or bone marrow-derived macrophages or monocytes that are circulating in the blood, and you put them directly into the brains, they cannot recapitulate that phenotype nicely. And primary of which is this, again, this P2RY12 receptor. So this receptor seems to confer on microglia some unique identity, and we are very interested in this receptor. This receptor itself is a purinergic receptor. That is, it responds to purines like ATP, and it's expressed on the surface of the microglia, both in their finger-like projections as well as on their cell bodies. It facilitates the response to ATP. Specifically, it facilitates the response to ADP, that's a breakdown product of ATP. And when it binds to the P2RY12 receptor, which is a G protein coupled receptor, it leads to downstream signaling that actually reorganizes the actin cytoskeleton and allows and facilitates the movement of these microglial cells. We know that about this receptor, but there's perhaps a lot more that this receptor does that we're still in the process of finding out. Given its unique specificity to the microglia in the brain, we decided to look at potential developmental expression of this gene. So this was done by a postdoc that's now in the lab about a year now. Dr. Kenneth Okoje comes to us, having been trained in Nigeria, completed his bachelor's and then his PhD, and then he joined us last year. And he started to ask the question over the course of development, across sex, what is the expression of this P2RY12 gene? And he shows from P7 all the way to P28 in the developmental frame, although there's an increased expression of this gene, there's no differences across sex between males and females up to P28. So during the developmental paradigm or context, there's not a lot of changes. However, when we look in the adult, what he sees and shows here is that this receptor is actually more highly expressed in females than it is in males. We're still wrestling with what that means or what that signifies, but it suggests that in the developmental paradigm, there's no difference at the sex level between these in the expression of this gene, but at the adult level, there's some difference. Now, a grad student in the lab, Joe Ware, who's also from Nigeria as well, and he's now in his 4th year, actually got me first interested in these sex differences, because I'll be honest, before Joe's data, I wasn't convinced that there were potentially sex differences, especially in the microglia. But Joe joined the lab, and during his before he joined the lab, he did a rotation with Dr. John Lukens, who's here at our center as well. And since I'm not trained in behavioral studies, he had to pick up the behavioral training from Dr. John Lukens. And for his thesis project, what Joe is going to do is starting to look at the behaviors that potentially may be regulated by this P12Y12 gene. What I'm going to do here is I'm going to summarize the data broadly and then present the specific results from that data. What is remarkable is that in relationship to some of the behavioral disorders that I mentioned earlier that are common and sexually dimorphic between males and females, Joe finds that in simply knocking out this one gene or one receptor that's present on the microglia, he can elicit behaviors that are reminiscent of anxiety behaviors and hyperlocomotory or ambulatory behaviors, specifically in female mice. In male mice, he doesn't see these phenotypes, but conversely, he sees all the phenotypes that show that the microglia P2Y12 may regulate social behaviors that are more commonly defective in males and communication behaviors that are also more commonly defective in males. For these studies, again, he used 3 to 5 month old wild type and knockout mice that were of both sexes. Again, that's what that shows there, wild type and knockout mice. And I'll start with showing you some of his data on females. So Joe used this elevated plus MACE test, and this test is a test that's known to be useful for testing anxiety. For context, mice are very social creatures, and they like to explore the environment, as well as they have an inhibitory behavior of being reserved and confining themselves more to enclosed spaces. So this elevated plus maze is a maze that's elevated and has this cross, and so that's why it's called a plus. There are two arms of the maze, what is called an enclosed arm that has walls, and then an open arm that doesn't have walls. Mice would like to explore the new environment, so typically they would spend a lot of time, especially in the enclosed arm, because that's comfortable. But sometimes they would also explore the open arm. Mice that are exceedingly anxious would spend little to no time in the open arm because they're more anxious to get in there. And so Joe did these tests on 3 to 5 month old mice. And first of all, in females, what he saw was that the females spent less time in the open arm, suggesting that they have increased anxiety. And the total distance that they traveled in this more restricted space was not different. When he looked at males, he didn't see any differences between the amount of time they spend in the open arm between wild types and knockouts. So this suggests that they have increased anxiety behaviors. He moved on to use the open field test, which is, again, a really open arena for mice to go ahead and explore. And also it can be used as a readout for their ambulatory or locomotory activity. And so Joe again used female mice, and what he saw was quite remarkable, that in the mutants, they showed an increased total distance that's traveled. So in the given period of time, in this case, it was about 10 minutes, these mice that lacked the receptor and were female were moving around the cage a lot more. They seem to show this hyper ambulatory behavior and impulsive behavior, such that they spend a lot more time crossing through the center of the space, the open arena, and they have a shorter period of time of latency. Once you put them in the cage in the corner, they like to go straight to the center. So they seem to have this impulsive activity where they're constantly active, almost like they're pacing around without stop. This is what he saw in females, but when he looked at the males, he could see no differences between the wild types and the knockouts, again suggesting that these mice have increased anxiety, and they also have increased locomotory behavior. The next thing Joe did was to try to move from the behavioral level to now ask at the circuit level what regions of the brain or what circuits in the brain are affected. And this is still ongoing work, but what he sees is actually quite interesting. In this case, we do the hyperlocomotion test, again, expose the mice to the open field for about 45 minutes. We have a cohort of mice that are not exposed to this, so in the naive condition. And in this case, we're looking at only female mice at this point. And then he looked at CFOS. CFOS is a marker for neuronal activity. So it is increased when neurons are active. And when he looked in the naive condition, he didn't see much of a difference between the wild type and the P12Y12 knockout mice. However, after exposing the mice to an open field, what you can appreciate in this context is that there's a lot more CFOS activity, showing that there's a lot more neuronal activity in wild-type mice. But in the mutant mice, the level of activity is significantly reduced, and that's quantified here. And this reduction is specific to the striatum, which is a specific region in the brain that's actually known to regulate motor activity. And again, this is present in the striatum, but if we look in other regions of the brain, like the hippocampus, or in this case, the ventral dentate gyrus region of the hippocampus, we don't see a difference in the level of CFOS that's expressed. This reduction in neuronal activity is not because there's a reduction in the total number of neurons in the striatum, because the total number of neurons is identical between wild types and mutant mice. So we have the same number of neurons, but despite the same number of neurons, when exposed to this open field activity, we see fewer active neurons in this context. So we've honed in on this specific striatal circuit that seems to be affected. What Joe wanted to look at was to also potentially identify specific cellular defects in the microglia, since behavior is affected and the circuit at the striatum seems to be affected. What about the cellular structure in the striatum of these microglia? Microglia, again, are these ramified cells, as you can appreciate from this image, and wild-type microglia are present here, and knockout microglia are present on the right. When he looks at the features of this, he observes that there's a significantly greater density of microglia in the mutants compared to the wild type. And this significantly greater density is evidenced also by the shorter average distance to the nearest neighbor. So these cells are more compacted together because there's a lot more of them in an identical space. Morphologically, we also find that these cells are significantly perturbed in their ramification, or how branched they are. They have a smaller soma size. They have a reduced ramification index. And the average branch length and the max branch length is also significantly reduced in these mice. So we see some perturbation by a loss of this P2R-Y12 receptor on the microglia that confers aberrant cellular phenotype that we think may translate to the circuit defects and then subsequently to the behavioral defects. And we're going to be pursuing that to look at that some more. But I just walked you through the female-based behaviors. And now I am going to walk you through the male biased behaviors that we also observe in these mice. Joe also did a group of other tests, including this dissociability test, which is a test of the animal's ability to be social. Mice are quite social creatures. They like to interact with other mice. So if you put them in a cage, if you put them to interact with another mouse or to interact with an object, they would rather spend more time with the other mouse. But when we looked at these in male mice that are known to show more social deficits in the human population, we see that while wild-type mice spend a lot more time with the unfamiliar mouse, the P2R-Y12 knockout mice shows no preference over this social interaction with a mouse versus an object, suggesting that these mice are actually deficient. I don't show you the data here, but we actually look at this in females, and we don't see these differences in females. Second, this indicates that these mutant mice have a social problem. But as a readout of the social problem, or something that could be responsible for the social problem, it may be a communication problem. And in the human population, this is also quite common, that a lot of times, especially, for example, in the case of autism, children who have social defects also have communication defects. So Joe tested this out in a specific assay to look at the vocalization, which is a way for mice to vocalize and communicate with one another, of males to an estrus female that he anesthetizes. He puts a microphone present there, listens to the mouse vocalizations over the course of 15 minutes, and then exposes the mouse to a female, and then listens to their vocalization over the course of 3 minutes. And here's what the data looks like. In the basal condition, what you can appreciate in wild-type mice is they have three, what we may call syllables, three patterns of vocalizations, or in the habituation period. But once they're exposed to the estrus female, those three get morphed in such a way that the split gets reduced and the step gets significantly increased, and the rise is a new syllable that's expressed while the wave is lost. And if you look at the mutant mice, we already see that the mutant mouse in the basal condition already starts to show features that seem similar to the already hyper-vocalized state of the wild types, where you can see these 3 syllables matching there. And once we expose it to the estrus female, we can actually see it develop 2 new syllables, the rise and then the wave, and also the inverted U, indicating that these mice actually have an altered vocalization behavior. Well, we can also quantify this in terms of the frequency variance of their communication. So it suggests that simply lacking this receptor, specifically in males, actually results in an altered form of communication in these mice. We went back to having seen these behaviors, we now again go to the circuit and try to narrow regions of the brain that are perturbed in these P2Y12 deficient male mice. And again, we expose them to the social activity. We look specifically in the prefrontal cortex, which is a region of the brain that's important in social interactions. And here we see that in the males, the density of CFOS is actually significantly reduced. Again, CFOS being a readout of neuronal activity, suggesting that in these mice, when they are exposed to social engagement, they actually have few neurons that are active. And so there's an hypofunction in these mice. Finally, we go on to the cellular, and like we did with the females, we look specifically again in the prefrontal cortex at the microglial density and morphology. In terms of density, we see a similar thing like we saw in the females, where these mice have more microglia present. However, in terms of their structure, we don't see a significant difference in the morphological structure between these mice. So to summarize what I've shown you so far, I show you that there's a sexually dimorphic expression of P2Y12 in adulthood specifically. And then what we see on both male and female angles is that, especially in the females, we see a hyperlocomotion and increased anxiety that we think is regulated by hypofunction in the striatal circuit. And that itself is regulated by the perturbation in the microglial density and morphology in females. In males, we see social and communication alterations that is regulated specifically in the prefrontal cortex and hypofunction in that region that we think is a result of the microglial density, but not their morphological perturbations. And finally, in terms of our future direction, what we'd like to look at is we'd like to clarify if these phenotypes that we observe, especially in terms of behavior, are specific to the microglia. I've mentioned to you that P2Y12 is specific to the microglia in the brain, and these behaviors we know are regulated by the brain. But P2Y12 is also present in other cell types outside the brain, and we're using a generic knockout mice. So we'd like to use approaches where we can conditionally knock out the receptors specifically in microglia in the brain and see if we can recapitulate those phenotypes. In terms of the circuits, we would like to determine the specific circuit dysfunction in females in the striatum and in males in the prefrontal cortex, and then also to look at the transcriptional profile of alterations lacking this receptor specifically in microglia, and even translate that to look at it in neurons as well. When microglia are perturbed, what's the neuronal profile when we lack this receptor? And with that, I would like to appreciate the lab. This is several of us in the lab. I especially talked about work that was done by Kenneth Okoje, and especially work that's ongoing right now for his thesis project, Joe Uweru. And I didn't have the time to talk about other members of the lab and their work. Maybe I'll get a chance to do that in future. We're present in the Department of Neuroscience at Virginia and in our Brain Immunology and Glia Center, where we look at the interface of the immune system and the brain. And this is our funding sources, both external, internal, as well as some that have been completed and some that are ongoing. And I'd like to end with an appreciation of my family that helps me to do the work that I do here, including my wife and our 4 kids. And I'll be happy to take any questions at this point. Thank you.
Thank you, Dr. Eyo, for a really great talk on how microglial purinergic receptors may regulate circuitry and behavior in a sex-specific way. As an immunologist, I can certainly enjoy all data that shows immune cells regulate everything. But it was really fascinating to see the effects on actual behavior. So we do have some questions in the chat. One person was asking, is the difference in the male versus female expression maybe due to a paring back of the receptor later in development? Do you think the use is less in males, or do you have any kind of hypotheses on that?
Well, that's a great question. We don't know. We don't know. We'll have to test it out. There are all sorts of potential reasons, right? So it could be like the questioner suggested that there's a reduction of use in males, but it could be that there's an altered use in males versus females and vice versa. There's still a lot of work to do here that we have to work that out.
And with the smaller, you saw a smaller size, like an increased number of microglia, but at least in the female, smaller size and branch length. Do you know, does that mean they're less active?
Well, so this is the big question. This is the big thing with in our microglia field. The word active or activated is means different things to different people. So I'm just describing the phenotype. I'm not labeling it active or not active, because technically microglia are always active, right? Maybe they are alternatively active in some way. So we don't know what that means. We don't know what the cell body size entirely means. We'd like to figure that out.
We have a couple questions about hormone regulation. So do we think the sex dimorphism could be regulated by hormones? And if so, could you use a model like the OVA-X type?
Absolutely. This is why we'll be busy in this field for a while, because there's still a lot of questions. I'm not trained in the hormonal regulations, but we're starting to collaborate with people to start to investigate that. Great suggestion.
Do you think that the anxious behavior and higher vocalization could be to compensate for the lower sociability? So can microglia only regulate sociability, but maybe cause the phenotype?
That's a fascinating question. And I think that there's quite some truth to that. That is because what I don't know is which is the chicken or the egg, right? I don't know. So I think that what is happening in the males is that they are problematic in their social activity, but that might be a result of the fact that, or because of their problem in social behavior, their vocalization is altered. So, for example, one of the things we're working on to look at is perhaps their auditory function is bad. And if they can't hear well, then they can't vocalize as well. And if they can't hear well and they can't vocalize as well, maybe that's why they don't interact as well. So there may be all of those things playing in together. Or it may be the other way around, that just because they have problems with socializing, then they don't want to communicate as much, right? So, but we don't know which one is the cause of the other. And we'll have to do some of these potentially specific. Once we know the circuits, then we can target it specifically to determine which one goes bad first.
That makes sense. Do you know if there's any alterations in the number or morphology of the neurons themselves?
So far, we haven't found any alterations in the density of the neurons. That's the number of neurons. Again, I showed you that in the new end, they have the same number of neurons. What we're starting to do now is to look at the synapses present on the neurons, and we can find alterations. I don't think it's ready for prime time yet, so I'll keep that to the student in our labs, but we can start to see some alterations there. We also haven't looked at the profile, so that's another thing that we want to do. We want to do the RNA-seq to see what's the neuronal profile like in these guys. We're pretty sure that they must be altered, but we'll have to describe and define what that alteration is like.
And then last, we have a question. Does this work look like it could be translational in any way to clinical trials? Do you have ideas about that yet?
Yeah, so of course, we would like to, we're in a school of medicine, we'd like to go there. Again, I'm not a clinically oriented person, but let me tell you what my thought is. So I don't show you the data. Some of the communication problems that we saw, that I mentioned to you, we actually see that only in adults. We don't see it in neonates. And what is interesting about that is, again, I'm dreaming here, so forgive me if it's not well thought out. But in patients that have, for example, in autism, where there are communication deficits and there are these social deficits, right? We sometimes catch the problem late, and the kids are already grown, and we can't do anything about the past. Well, if potentially P2RY12 could be a target that we can use to modify the system and improve things, this could have application to the clinic where we can target it, not worrying about what happened in the past, but we can modulate it now to improve behavior. Again, there's still a lot of work to do with that. We'd have to modulate P2Y12 in adulthood and see that actually changes potential behaviors. Perhaps in these autism models, we can modulate it. But that would be some of the directions that we'd like to go to see if we can target. And there are actually drugs that are already available and present to target this receptor.
That's really exciting. I see Dr. Alfaki is in the chat answering some of the other questions. I might send you there as well because there's a whole lot of questions related to your work.
All right.
Lots of people are interested, so thank you so much.
Thank you very much. Thanks for the invitation. I appreciate it.
So next we're going to have Dr. Sara Suliman. Dr. Suliman is an immunologist by training with a focus on understanding the risk of progression to active disease and developing predictive biomarkers. She's currently an assistant professor at the Division of Experimental Medicine at UCSF and the Zuckerberg San Francisco General Hospital. She was an instructor at the Brigham and Women's Hospital and Harvard Medical Schools, where she focused on dissecting host factors associated with TB risk, including genetic associations and T-cell responses. She also led the COVID-19 Diagnostics Accelerator Lab under the Mass General Brigham Center for COVID Innovation. Welcome, Dr. Suliman. We can see your slides, but I believe you're on mute.
Of course, I'm on mute. Apparently, this was the most spoken phrase over the last year. So can you hear me and see the slides okay?
Yes, I can.
All right. Thank you so much for the opportunity and privilege really to speak at the Black in Immuno week. And I just wanted to give a shout out to all the organizers for the amazing work they've been doing since last year to create this platform for Black immunologists. So I work on tuberculosis disease, and as of 2019, at least, before COVID, it was the highest infectious disease killer. And my work focuses on systems biology approaches that integrate genetic and transcriptional profiles to understand new mechanisms of TB susceptibility. So just by way of introduction, the global burden of tuberculosis is still very high. And because of all the interruptions to the TB services over the past year, because of the COVID pandemic, we've actually seen a rise in the number of deaths and incidents in TB. We can see that there's a high cluster of TB incidents in sub-Saharan Africa because of the co-pandemic with HIV. And there's still several areas in the world where there's a high incidence of TB. And the main issue that we think about in TB is that there's a very large reservoir of individuals that are latently infected with Mycobacterium tuberculosis. So that's the bacteria that causes TB disease. And the estimate is 1/4 to 1/3 of the world is actually infected with MTB. So one of the main questions in the field is why the majority of individuals that are exposed to TB never actually develop TB disease. What's special about the individuals that are exposed? So what you can see here in this diagram is the life cycle of TB disease. You start with exposure to the bacterium, MTB, and the estimate is that 5 to 10% of individuals who have been exposed to MTB have a lifetime risk of developing active TB disease. So this leads to several hypotheses, and especially since hosts would be infected with similar strains of circulating Mycobacterium tuberculosis, it leads to the hypothesis that host-specific risk factors, including genetics, might explain this difference between progressors and non-progressors. So one way to address the genetics hypothesis is by conducting genome-wide association studies. And GWAS studies are studies where you enroll cases and controls that are matched by age, gender, and other similar other characteristics. And you genotype these individuals, so you profile all the variants in the genome. And what you'd like to see, this is an example where allele A, for example, is associated with a higher risk of disease. So you see an enrichment of individuals with the AA genotype among the cases. whereas the C is associated with protection. So you see an enrichment with individuals with the C genotype in the controls. And you do this over the entire genome and identify genetic loci that are associated with high risk of disease. The added layer of complexity when you have an infectious disease GWAS is that you also need to control for exposure to the pathogen, in this case, Mycobacterium tuberculosis. To address this hypothesis, I worked with Megan Murray, who's a professor at Harvard School of Public Health, where she led the design of this household contact study in Lima, Peru. You can see this is a high density area where TB transmission is high, and you can control for exposure by conducting a household contact study where you can actually follow up individuals who have active TB disease and their contacts, so you know that they have been exposed to the bacteria. So the parent study design goes as follows. We have over 3,000 index cases. So these are the individuals that we identify as the initial source of the infection, and we follow the household contacts. And the household contacts that are tuberculin skin test negative, so they don't have evidence of exposure to MTB by virtue of their skin test result, are followed up for a period of 18 months. And individuals who converted from a negative to a positive skin test have a confirmation that they have been infected with MTB. So those are the controls in our subsequent GWAS study. And for the definition of the cases, we include both the index TB cases and the secondary contacts. So the Genome-Wide Association study has been published in Nature Communications, and it basically addresses the hypothesis that host genetic variation might confer susceptibility to TB disease. So I worked with Yang Lu, who's now a junior faculty at the Broad, and she conducted this Genome-Wide Association study on Peruvian household contacts, where we enrolled about 2,000 cases and 2,000 controls. And Yang identified this non-coding locus on chromosome 3, where there was a cluster of genetic variants that are associated with progression to TB disease. It's a modest odds ratio, which is expected for an infectious disease GWAS, but the lead SNP was this SNP on chromosome 3, where the A allele, which is the minor allele, was associated with higher risk of progression to TB. So since this is in a non-coding variant, how can we infer some mechanistic insight into that? One mechanism by which non-coding regions can confer susceptibility to disease or regulate gene expression is through the following mechanism. So we have an example here of uninfected dendritic cells where you have no transcription factor binding sites because the uninfected cells don't express the transcription factor of interest. But once these TCs are infected with MTB, you can see some chromatin remodeling as well as transcription factor expression. And due to the genetic variation at the transcription factor binding site, you can imagine a scenario where the C allele in this example has lower transcription factor binding sites, whereas the A allele has higher binding, which results in upregulation of gene expression. The paradigm is that the non-coding variants would regulate gene expression by virtue of acting as enhancers or promoters. But then the question becomes, which cell type do we actually target for identifying these regulatory events? So I worked with Tiffany Amariuta, who developed this algorithm called IMPACT, or Inference Modeling of Phenotype-Related Active Transcription. And what she did is survey a number of epigenetic and genetic data sets where she identified genetic regulatory features and identified over 500 different genetic features from public data sets. And based on how well these genetic regulatory features predicted transcription factor binding data sets in ChIP-seq data, she would assign an impact score from 0 to 1, 1 being the highest. That indicates that there's a predictive power of these genetic regulatory events of transcription factor binding. And because transcription factors tend to be associated with specific cell types, or they can be actually master regulators of different cell types, we can infer the cellular identity where these regions could be active. So she applied the impact algorithm on the non-coding region that we identified in the Genome-Wide Association study. And you can see this high impact score, which predicts transcription factor binding for a monocyte-specific transcription factor, CEBPP. So this led to the hypothesis that our lead SNP marks a monocyte-specific enhancer. So for me to address this question in the lab and try to validate the interaction between the enhancer and gene regulation, I used the CRISPR-Cas9 system, where basically used the Cas9 nuclease to introduce a high number of double-stranded breaks. And since the cells would try to repair these double-stranded breaks, it would introduce a high number of insertion deletions. So the idea here is to introduce a high number of insertion deletions that would break the conformation of the enhancer, and then identify which transcripts are regulated as a result of that. So the prediction is that the disruption of the enhancer would change gene expression and give us an insight into what the candidate risk gene would be. So I did this experiment in THP1 cells, which is a monocytic cell line, where I introduced 3 single guide RNAs to introduce a high number of insertion deletions around the lead SNP, and then sorted single cells from these bulk THP1 cells that have been edited, and analyzed them by both Sanger sequencing to analyze the genomic sequences and RNA sequencing to identify the transcripts that are potentially regulated by this disruption. So this is just a snapshot of some of the clones. So the ones in blue have the same, have not been edited, so they have the same orientation and sequence as the wild type clones. But then the ones that are coded in red would have a high number of disruptions. So you can see either that they have the same size as the wild type clones, but there's a number of edits, or they have large deletions. So you can see this depicted here in this gel, where I PCR amplified the region around the edits, and you can see that some of them have very clear deletions, whereas some of them have the same size as the wild type clones, but have an edited sequence. So I performed this RNA sequencing experiment where we compared the wild type and the edited clones. And this was probably the most expensive negative result I ever generated. So you can see there's no difference between wild type and edited cells. And this is actually a common problem in genome-wide association studies. It's very difficult to link the variants to gene transcripts. So then I looked into other data sets to help identify what the gene of interest would be. So I worked previously at the University of Cape Town with this African household contact study, where we followed up index cases and household contacts in 4 different African countries. And in a very similar design, we followed up the contacts of these index TB cases. So this is a different representation of the study design, where we start with household contacts of TB cases, and then follow them up for 2 years. So some of the individuals would develop TB disease, and you can see the matched controls on this side. So then what we do is align people by the time of TB diagnosis, and then retrospectively analyze samples that have been collected from these individuals, and perform RNA sequencing on whole blood samples. So this would give us an actual insight into prospective transcriptional changes before people develop TB disease. So the workflow starts with these frozen PAX gene tubes, where we extract RNA samples and perform global RNA sequencing. And the initial purpose of the study was actually to develop biomarkers. So we translated the most predictive genes into qPCR data, but we can also use these whole transcriptome data sets into gaining insight into mechanisms of progression to TB disease. So I worked with Fergal Duffy, who actually analyzed the modules that were regulated by as a function of progression to TB, and we found this module that has a high number of genes that regulate ion transport. And also looking at chromatin capture data sets online, we can see that our lead SNP is actually physically associated with the promoter of ATP1B3. So now we have 2 examples, one from a household contact study in sub-Saharan Africa, and one from a public data set that suggests that our variants that are identified in Peruvian study participants is potentially associated with ATP1B3. So that leads to the hypothesis that this is a candidate risk gene for TB progression. So what is ATP1B3? It's a sodium-potassium ATPase, and this is the transporting subunit beta-3. There are 4 alpha isoforms and 4 beta isoforms and a regulatory domain. And this is an active process that requires ATP hydrolysis to maintain a high extracellular sodium concentration and a high intracellular potassium concentration. And looking at public data sets of monocytes that are infected with MTB, we can see that in acute infection models, we see a clear upregulation of ATP1B3. So in order to analyze the function of ATP1B3 in MTB infection, I generated THP1 clonal cell lines that have been edited for ATP1B3 or TCR alpha constant region as a control, and then sorted ATP1B3 sufficient and deficient cells. And the prediction here, since MTB infection leads to higher ATP1B3 expression, that MTB growth will be lower if we infect knockout cells. So this is just sorting the ATP1B3 deficient and sufficient cells from the bulk edited THP1 cells and then expanding them for 6 weeks. And we can see that we still maintain the positive and negative clones based on ATP1B3 expression. So then we use this model of MTB where we can detect MTB growth by luminescence and infected these ATP1B3 sufficient and deficient cells. And what you can see here, this is very preliminary and very exciting as well. You can see that in the cells in red, that ATP1B3 deficient cells indeed had lower infection or lower burden of MTB based on the luminescence compared to either the wild type cells or the cells that have been edited with the irrelevant guides that target TCR alpha. So to give you another example of formally integrating variants and gene expression, we also used the subset of participants that was re-recruited from the parent Genome-Wide Association study. So I mentioned that we followed household contacts of index TB cases. And then 5 years later, we re-recruited some of the individuals who stayed latent and never developed active TB disease or the TB cases. And what we're doing here is integrating the genetic variation with RNA sequencing data. So for that, we enriched monocytes that express CD14 and either differentiated them with GM-CSF and IL-4 to dendritic cells or with MCSF into macrophages. And since this was nested from the GWAS study, we have the genotyping information for all these individuals. And now we have the low input RNA sequencing data from both the monocyte-derived DCs and macrophages. So we can integrate the two data sets into an EQTL study, which I will explain next. So just as a first pass quality control, we just compared the two cell types to ensure that they express the lineage specific markers that we expect. So we can see here in the DCs, they express the genes that we expect them to express, like chemokine ligand 17 and 22 and group 1 CD1 proteins. And the macrophages express the genes that they're expected to express, like maintaining a high expression of CD14 and expressing CD163 and Fc gamma receptors. But we found no TB association. So if you look at the differential expression within the DCs and macrophages, there are no genes that are specifically regulated in progressors versus non-progressors, which is not unexpected since they were re-recruited 5 years after the initial study, and also the in vitro differentiation might change their transcriptional profiles. But now we have an opportunity to conduct an expression quantitative trait loci study. So the idea of EQTLs is that you run these linear regression models where you integrate the genotyping data with the expression of different genes. So you can see an example where there's no association between the genotype and the gene expression on the left. But on the right, you can see that if you have a T allele, then you have a higher expression of your gene of interest. And this could be due to disruption of specific transcription factor binding sites, for example. So we have this data set from DCs versus macrophages, and also we have an opportunity to analyze this in cases versus controls. And I worked with Maria Gutierrez Arcelas, who's now a junior faculty at Boston Children. And we identified first a cell type specific EQTL events. So in DCs, we found 433 significant interactions and 355 in macrophages. And of those, there were 79 that were specific to one cell type, but not the other. So this is the top macrophage associated interaction, where you can see that Gasdermin A was associated with the genotype in macrophages, but not in DCs. And CD1A was the top DC-associated gene, where you can see that the minor allele was associated with lower expression of CD1A in DCs. So to analyze the interaction with TB, we applied this model where we regressed the expression on the genotype, the TB status, a number of other variables, and also the interaction between genotype and TB. And the top hit was this gene, FAH, where we found that in people who developed TB disease, so the progressors, there was an interaction between the T allele and lower expression of FAH, which we did not detect in the controls. And in macrophages, we did not detect this association. So this was both a cell type specific interaction and also a disease specific interaction, where the progressors showed regulatory interaction, but not the non-progressors or the controls. So what is FAH? It's femoral acetoacetate hydrolase. It's the last enzyme in tyrosine metabolism. And what it does is break down femoral acetoacetate into fumarate and acetoacetate. And just looking at the literature, we found that FAH loss of function mutations were associated with hereditary type 1 hypertyrosinemia, so accumulation of tyrosine in the blood, which is expected. And again, analyzing public RNA-seq dataset, I found that acute infection of monocyte-derived DCs with MTB resulted in lower expression of FAH. So this is an exciting hypothesis, and it leads to the idea that tyrosine metabolism is regulated in the context of MTB infection and could be associated with TB disease. So in summary, I showed you evidence that integrating different types of data like genetics and transcriptomics, either from the same study or from different studies, can provide novel insights into the mechanisms of TB progression. So I've shown you an example where we identified the candidate pathway in sodium-potassium ATPases. And another example where EQTL studies using genetic and transcriptional data from the same participants led to the hypothesis that tyrosine metabolism was associated with TB progression. With that, just wanted to give a quick announcement here that my lab has opened. Now it's been 6 weeks, and I'm actively looking for candidates. So if you haven't applied to, if you're looking for grad school opportunities, I encourage you to apply to the Biomedical Sciences graduate program where I'm affiliated. The application deadline is December 1st, so you still have 2 weeks to put your application together. And if you're interested in other opportunities, please contact me, and we can definitely talk more about that if you find any of this work interesting. And with that, I would like to acknowledge my former mentors when I was at the Brigham, Branch, Ildiko, Shomo, Megan, and a number of people that I've acknowledged as we went through the presentation, and also the African Consortium, where we conducted the whole blood transcriptional analysis. And thank you for that.
Thank you, Dr. Suliman, for a great talk investigating the potential causal factors for the development of TB disease upon exposure. As a trainee, I really appreciated you showing some of that negative data just to normalize the struggles that I'm sure we all have had or will face at some point. And we definitely have questions from the audience for you. Juliette Irene Murillo Silva said, can you have used monocyte-derived cells from the people genotyped in Peru to evaluate differences in gene expression in the MTB-infected cells?
Yeah, so that's an interesting question. So our initial design for the study was actually to include an arm where we infect the cells and then conduct the transcriptional profiling, but then the samples were limiting. So unfortunately, we don't have that information upfront. But that's an interesting hypothesis. And I actually want to take it a step beyond the genotyped cells and actually edit these genes in primary monocyte-derived DCs and macrophages and identify whether that has an impact on MTB growth.
Yeah. Neil Silverman asked, did you try stimulating THP1 cells or others and then the RNA-seq, for example, with LPS or dead or alive, TB, cord factor? Have we done any stimulation experiments?
Yeah, Neil, that's an excellent question. And I actually get this quite a lot after actually performing the RNA-seq data set and presenting this data. So sure, like one potential explanation for the negative finding is that the cells were not in the right context. So maybe we needed to stimulate their TLR ligands or TLR receptors or infect them with MTB to actually get at the interaction. But there's an unlimited number of options for the cell context that we have to actually identify the regulatory event. So it was exciting. It would have added more conditions to the RNA sequencing experiment, but then it still had the same risk of not providing a clear result.
We always wish for more samples, right?
Yeah.
Katrine Meyer-Barber asked, she said, wonderful talk. Do you think ATP1B3 could regulate ATP to modulate inflammasome activation?
Hi, Katrine. Nice to see you here. So actually, the working hypothesis is that ATP1B3 deletion would increase potassium efflux, which is a trigger for inflammasome activation. But I just started also thinking about the ATPase side of the equation, because that is also an inflammasome activation trigger. So I'm happy to talk more to you offline about ideas about how to proceed forward with the project.
And then our first speaker, Dr. Alfaki, said, are there examples from the literature about tyrosine metabolism in infectious disease or in TB in particular?
Yeah, that's an excellent question. I actually looked through the literature or case studies of people who have the hereditary type hypertyrosinemia, but it's a very rare genetic disease, and I couldn't find any data that describes susceptibility to infection, mycobacterium or otherwise. That would have been an interesting association and would definitely strengthen the hypothesis that regulation of tyrosine metabolism is associated with infection. There's a lot of data on other amino acids and their role in MTB. There's an idea that nitrogen stealing from the host would reduce the available nitric oxide in macrophages. So then they're taking away the amino acids as a way of diverting the host from being able to generate reactive oxygen species that would kill the MTB. So that's one interesting hypothesis, but it may just be, a way of limiting the host ability to metabolize tyrosine.
That's great. Yes, so much work to do, but it's all really exciting. And good luck with your lab.
Oh, thank you.
I also want to shout out all the speakers. They're hopping over to the audience platform to answer questions. I didn't realize we couldn't do it from backstage. So that's extra work on their side, which is really appreciated. Well, thank you, Dr. Suliman.
Thank you.
Last but not least, we'll have Dr. Zuri Sullivan. Dr. Sullivan is an HHMI Hannah Gray postdoctoral fellow in the Department of Molecular and Cellular Biology at Harvard University. Her work in the laboratory of Dr. Catherine Dulac aims to define mechanisms by which neuroimmune interactions regulate sickness behavior. Dr. Sullivan conducted her doctoral work in the laboratory of Dr. Ruslan Metsitov in the Department of Immunobiology at Yale, where she defined a novel role for gamma delta T cells in the intestinal response to nutrient sensing. Dr. Sullivan is the recipient of numerous awards and honors, including the HHMI Hannah Gray Fellowship, the Yale University John Addison Porter Dissertation Prize, the NSF Graduate Research Fellowship, and a Fulbright Scholarship. In addition to her scientific contribution, she has led numerous initiatives aimed at increasing scientific literacy among the public, which is extra important right now, where we're realizing the importance right now. And increasing diversity and inclusion within academia, both at the institutional and at national scales. Welcome, Dr. Sullivan.
Thank you so much, Heather, and thank you to the organizers for the opportunity to speak today. Black and Immuno meant so much to me last year when it was founded, so it's really an honor to be presenting to this audience in particular. So as Heather mentioned, I am a postdoc at Harvard, and I currently work on neuroimmune interactions, but today I'm going to be speaking about my graduate work that I did in Ruslan Medzetov's lab at Yale University, where I was focused on the gut, which is the beautiful organ that you see pictured on this slide. So I want to begin with this pair of videos that at first pass look unrelated, but they actually depict the same thing. So these are two animals that are eating, and they do this using their digestive systems. And eating actually is a defining feature of what makes us animals. We're unable to produce our own food like plants, and so we rely on harvesting energy from our environments, which we do using our digestive systems. And so although animals take very diverse forms, from this trichoplax on the left to this great white shark that you see on the right, the one thing that we all have in common as animals is that we use our digestive systems to harvest energy from our environments. And so this is why this is a system that I find particularly fascinating as a biologist to study. So in the same way that animals can take very diverse physical forms, our diets also range in diversity. And so we can think about animal diets as existing along a spectrum. So on one end, we have specialists like pandas that subsist almost exclusively on a single food source for their digestion. So in this case, pandas, of course, use bamboo as their primary food source. And on the other end of the spectrum, we have generalists like ourselves and President Obama, who can, of course, consume a variety of foods from day to day and throughout our lives. And it's interesting to note that the existence of these diverse diets over the course of evolution has given rise to diverse organizational structures in animal digestive tracts. And so for specialists like herbivores and a rabbit, for example, we can see that they have an exaggerated small intestine and cecum, which enables them to digest the complex polysaccharides that make up the majority of their diets. And this is in comparison to a carnivore of similar body size, which has a much smaller small intestine and cecum, and whose diets primarily consist of proteins and lipids. But what about for generalists like ourselves, whose diets change on a day-to-day and seasonal and lifelong basis? We can't have these fixed morphologic adaptations in the way that our digestive systems are organized. And so this really formed the foundation for the project that I'll be speaking about today. The idea that omnivorous lifestyles necessitate intestinal adaptability to diverse foods. And this led to the central question that I sought to answer during my PhD work, which is whether nutrient handling machinery is dynamically regulated in response to diet. And so what do I mean when I say nutrient handling machinery? I'll quickly walk you through an overview of what happens when we eat, just as a reminder. So when we consume a meal like this hamburger, we chew it in our mouth, we swallow it, it travels down to the stomach, where it lands in the lumen of the small intestine. And here it's first acted upon by enzymes that are secreted by the pancreas that break it down into smaller products. It's further broken down by enzymes that are expressed on the brush border of small intestinal epithelial cells. And then this yields products that are then absorbed across the apical layer of the small intestine epithelium, where all of these products can be used to fuel our many physiologic processes. So just as our diets consist of various different types of macronutrients, proteins, carbohydrates, and lipids, we also have specialized digestive machinery, both pancreatic enzymes as well as brush border enzymes and transporters that are specialized for each of these specific types of macronutrients. And so today I'm going to focus specifically on carbohydrate handling machinery. And so I'm going to zoom in just a little bit more on the carbohydrate digestion process. So carbohydrate digestion and absorption machinery is regulated as follows. Most of our carbohydrate digestion actually happens with our microbiota, to which we outsource a lot of this activity, but there are a lot of host machinery that can also participate in this process. So when we eat a meal that contains starch, for example, this enzyme called amylase that's secreted from the pancreas breaks this down into various oligosaccharide products such as maltose, maltotriose, sucrose. These are then broken down by these brush border enzymes that are expressed on enterocytes, lactase, which breaks down lactose, as well as sucrase isomaltase and maltase glucoamylase. And these yield these individual monosaccharides that are then absorbed across the epithelial barrier by these absorptive cells called enterocytes, and finally across the basolateral membrane, where they can again be used to fuel host physiology. And so the genes that encode these various brush border enzymes and transporters are what I'll be referring to going forward as the carbohydrate transcriptional program. And these are the genes that we wanted to understand whether or not they were dynamically regulated in response to diet. And so to test this question, we designed 2 diets that differed only in the ratio of protein to carbohydrates, and we fed them to mice for 5 days, after which time we harvested small intestine epithelial cells and looked at their gene expression. And so in this volcano plot here, you can see the results of this RNA sequencing analysis, in which transcripts that are upregulated in mice fed the high carb diet are on the right, and transcripts that are upregulated in mice fed the high protein diet are on the left. And the transcripts that correspond to nutrient handling machinery are colored to correspond to the particular macronutrient type that they digest. And so what you'll see when you look at this volcano plot is that the transcripts involved in carbohydrate processing are upregulated in the high carb diet. The genes that are involved in high processing protein are upregulated in animals fed a high protein diet. But if we look at the machinery involved in lipid handling, we see no difference between the 2 diets. And this makes sense because, as I mentioned, these diets are matched for lipid content, and the only difference between them is the amount of protein versus carbohydrates. And so what this showed us is that this nutrient handling machinery is in fact induced on demand, as we predicted. And because we saw this very strong and robust upregulation of the carbohydrate handling machinery in high carb diet, this is what we went on to focus on throughout the rest of the study to understand the mechanisms that underlie this regulation. So we confirmed that this carbohydrate transcriptional program was upregulated in the small intestine epithelium using quantitative PCR analysis. And interestingly, we also looked at gene expression in the pancreas and found that the genes that encode pancreatic amylase were also upregulated in high carb diet. And so not only is this on-demand induction of nutrient handling machinery present in the intestine, it's also present in the pancreas, the other part of the digestive system. So next, we wanted to confirm that the transcriptional changes that we observed reflect functional differences in nutrient handling. And so we took a few different approaches to do this. First, we tried to approximate the rate of glucose uptake in the intestine by gavaging mice with glucose bolus and then measuring their blood glucose concentration every 5 minutes after gavage. And so what you can see here is that the animals fed the high carb diet, which again show an increased expression of glucose transporters, also show a more rapid rate of glucose uptake from the intestine, which is what we would expect given the transcription results. Next, we quantified organismal metabolism using metabolic cages, and we can measure something called the respiratory exchange ratio. And this is the ratio of carbon dioxide production to oxygen consumption, and it's a proxy for fuel utilization. And so what you can see with these traces in the middle and then the quantification on the right is that there's an increase in the respiratory exchange ratio in animals fed a high carb versus high protein diet, again indicating that there are functional differences in nutrient handling that accompany the transcriptional changes that we observed. The next question that we wanted to ask is whether or not this carbohydrate transcriptional program induction was indeed due to the availability of polysaccharides specifically. And so to test this, we designed 2 new diets that are now matched for the protein content, but differ in the ratio of fat to carbohydrates. And we again looked at the expression of this carbohydrate transcriptional program. And what you'll see is that in the high carb fed mice, we still see an increase in the expression of this program independent of the presence of protein, which is matched across the diets. And then finally, we used a drug that's an alpha-glucosidase inhibitor called acarbose to block the availability of carbohydrates in our high-carb fed mice. And here again, we see that the results of this experiment indicate that carbohydrate availability specifically drives this transcriptional response because the mice that are treated with the drug and therefore have a limited carbohydrate availability show a reduction in the expression of this program. So having established that this is a robust model for understanding the regulation of nutrient handling machinery in the intestine, we went on to ask how this program was regulated. And so we came up with two non-mutually exclusive hypotheses with how this regulation might happen at the cellular and transcriptional level. So the first is that individual absorptive epithelial cells called enterocytes might upregulate this carbohydrate transcriptional program. And the second possibility is that there might actually exist specialized populations of enterocytes that deal with different types of nutrients, and that diet might actually alter the frequency of these specialized cell types. So to first get at this question, we performed sort of a pulse chase experiment where we fed mice the high carb diet for 5 days and then switched them to the high protein diet for 1 or 5 days. Because we would predict that if the hypothesis on the right were correct, it would take a bit of time for the transcriptional phenotype to revert. And that's indeed what we saw. So when we switched animals from a high carb to high protein diet for just 1 day, we see no difference in the expression of this program. However, after 5 days of being switched to the high protein diet, we now see that the program shows reduced expression. And so this supported the hypothesis of altered epithelial differentiation. But to confirm this, we performed single cell sequencing to actually look at the cellular composition of the intestinal epithelium. And so this was performed in collaboration with Aviv Regev's lab at the Broad Institute. We performed single cell sequencing of the small intestine epithelium, and mice fed either a high carb or high protein diet. And we observed all the typical epithelial subtypes that one would observe. But what I want to call your attention to is that we also identified two as yet undiscovered specialized subsets of enterocytes. So these were marked by the expression of fatty acid binding protein 1 and fatty acid binding protein 6. And when we looked at the frequency of these specialized enterocytes, which differed also in the expression of the carbohydrate transcriptional program, we see that the frequency of these enterocytes do change in response to diet. And so this indicates that within 5 days, diet can actually alter the cellular composition of the intestinal epithelium, which then gives rise to the transcriptional changes that we observe. And this is the first demonstration that diet can actually alter the cellular composition of the intestinal epithelium, as well as the first identification of specialized epithelial cell types that deal with particular nutrients. And so this was a really interesting result for us. And so we went on to ask more about how this might be regulated at the tissue level. And so we can think of 4 different modes of epithelial sensing that are identified in the literature that might explain the phenotype we observed. So there's sensing that occurs at the level of individual absorptive enterocytes that sense an environmental trigger and autoregulate their own transcription. There are also specialized sensory epithelial cells that can communicate with neighboring enterocytes in response to environmental signals. There's communication between sensory epithelial cells and the nervous system through the vagus nerve, as well as the enteric nervous system. And finally, these sensory epithelial cells can communicate with immune cells, such as lymphocytes that are located in the tissue to regulate a tissue-level response to an environmental trigger. And so first, we tried to address these first 2 modes of epithelial sensing, the absorptive enterocyte intrinsic and epithelial communication. So to do this, we made use of an in vitro system called intestinal organoids, which are 3D culture systems in which we can grow all of the epithelial cell types and look at their gene expression in response to different perturbations. And so when we treated these organoids with different concentrations of glucose, we saw no difference in the expression of the carbohydrate transcriptional program. So this indicated that direct epithelial sensing was insufficient to induce this program and that other cells might be involved because all we have in our organoids were epithelial cells themselves. And so then returning to these 4 different modes of epithelial sensing, we particularly focused on this 4th mode in which epithelial cells might communicate with immune cells to regulate a tissue-level response to the environment. And this was interesting to us because during the course of my PhD, a number of groups had demonstrated that epithelial lymphocyte circuits regulate the tissue adaptation to intestinal pathogens. So when animals are infected with helminth or bacteria, specialized sensory epithelial cells detect the infection. Tissue-resident lymphocytes then amplify this detection to coordinate an epithelial remodeling response with the net effect of increased barrier defense at the tissue level. And so this has been well documented in the case of host defense. And so we asked whether lymphocytes might be similarly involved in the regulation of the intestinal response to different nutrients. And so we took an iterative approach to ask whether different lymphocyte populations in the intestine might be involved in regulating this carbohydrate transcriptional program. So first, we addressed the role of T cells and ILCs in the regulation of this program by treating mice with an anti-Thi1 antibody that'll deplete these two cell types. And we saw that this program did show diminished expression in the antibody-treated animals, indicating that either T cells or ILCs would be involved in the regulation of this program. Next, we used RAG knockout mice, which lacked mature T and B cells, and we saw a similar phenotype. And so these two results together indicated that a T cell was responsible for the regulation of this carbohydrate transcriptional program. Next, we used mice that were either genetically deficient in alpha-beta T cells or mice that that we treated with an alpha-beta T cell depleting antibody. And we saw no difference in the expression of this program, indicating that alpha-beta T cells were dispensable for the induction of the carbohydrate transcriptional program. However, when we used mice that lacked gamma-delta T cells or were treated with an anti-gamma-delta depleting antibody, we saw a reduction in this program, indicating that gamma-delta T cells are required for the epithelial response to diet. And that's summarized here. And this was a really interesting result because although gamma-delta T cells have been well studied for decades, we know very little about their biology, and even less about their homeostatic function in barrier tissues. And so this represented an opportunity for us to learn more about the biology of gamma-delta T cells. And so we undertook a number of approaches to understand how gamma-delta T cells are affected by diet. We used flow cytometry to look at how diet alters the phenotype of gamma-delta T cells. We used intravital imaging to look at how gamma-delta T cells tissue localization is changed by diet. We used RNA sequencing, bulk RNA sequencing, to look at how gamma-delta T cells transcriptome is altered by diet. And finally, we used single-cell RNA sequencing to understand how diet alters the population dynamics of gamma-delta T cells. So in collaboration with Daniel Masita's lab at Rockefeller University, we performed intravital imaging of gamma-delta T cells using gamma-delta reporter mice. And what's interesting about gamma-delta T cells is they exist in two developmentally and functionally distinct compartments in the intestine. And so we have the intraepithelial gamma-delta T cells, which interact intimately with the intestinal epithelial cells, and these are shown on the rings around the villi here. We also have the lamina propria gamma-delta T cells, which are much more restricted in number. And so what you can see in this pair of videos, which compares the intestine of a high-carb-fed mouse to a high-protein-fed mouse, is that there's a reduction in the intestinal epithelial fraction of gamma-delta T cells and an increase in the lamina propria fraction of gamma-delta T cells in the high-protein-fed mice. And this is perhaps easier to see in these 3D tissue-cleared images, as well as the quantification on the right. So next, we tried to understand how diet might alter the transcriptome of gamma-delta T cells to get a better handle on how gamma-delta T cells might in turn regulate the epithelial response to nutrient sensing. And so we performed RNA sequencing analysis of both gamma-delta and alpha-beta T cells isolated from the intraepithelial and lamina propria fraction of the intestine. And what I thought was really striking about this data is that there are very few transcriptional differences in the alpha-beta T cells, as indicated by the not very rich colors that you see in this heat map that corresponded to the fold change in individual transcripts between animals fed a high-carb or high-protein diet. And this is in contrast to the gamma-delta compartment, in which you can see there are a number of strong transcriptional changes that occur in response to diet. And this is particularly true in the lamina propria compartment. And I want to zoom in on one of the interesting results that came out of this RNA sequencing analysis, which is that we saw an altered expression of various components of Notch signaling in gamma-delta T cells, specifically in the lamina propria compartment in response to diet. And so depicted in this cartoon here are the various components of the Notch signaling pathway. And you can see that both the Notch 1 and 2 receptors, as well as Notch target genes, are upregulated specifically in gamma-delta T cells in the lamina propria compartment. And the reason this caught our attention is that we had also observed an upregulation of the Notch ligand JAGAD2 in epithelial cells in response to high-carb diet. And so this led us to speculate that perhaps Notch and JAGAD might be the mechanism by which the epithelial cells communicate with gamma delta T cells to regulate this transcriptional program. And the other component of this hypothesis was that perhaps there might be a change in the localization of gamma delta T cells along the crypt to villus axis in response to diet. Because we know that Notch and Wnt ligands are more concentrated at the base of the intestinal crypts, and so we speculated that perhaps gamma delta T cells might localize to the base of the crypts in order to sense the JAGAD coming from epithelial cells. And so to do this, we collaborated with Daniel Musita's lab again and performed 3D tissue cleared imaging. And so what you can see in this representation here is that there's indeed a higher concentration of gamma delta T cells more proximal to the crypt, where there's a greater number of Notch ligands. And so diet seems to alter the localization of gamma delta T cells, both between the intraepithelial and lamina propria fraction, as well as along the crypt to villus axis. And so next we asked whether this Notch JAGAD signaling might actually regulate the expression of this carbohydrate transcriptional program. And to do this, we treated mice with an antibody that'll block the JAGAD2 Notch ligand. And here we found that there was a reduced expression of the carbohydrate transcriptional program in the small intestine epithelium of mice treated with the antibody, indicating that JAGAD2 Notch signaling does indeed regulate this program. But through all of this analysis of our copious amounts of sequencing data, we were still unable to identify the specific mechanism by which gamma delta T cells regulate this program. And so to do this, we turned to the literature and focused on IL-22. And this is for a few different reasons. One is that gamma delta T cells in the skin are known to be a potent source of IL-22. Second is that it's also known that IL-22 can regulate the proliferation and gene expression of small intestine epithelial cells. And finally, that IL-22 can also regulate the phenotype of animals in diet-induced obesity models and can protect them from the negative impacts of high-fat diet feeding. And so given all this data, we hypothesized that in response to high-carb diet, gamma delta T cells might upregulate IL-22 and that this might upregulate the carbohydrate transcriptional program. And through a number of experiments that I don't have enough time to get into, we found that actually the opposite was true. So in response to high protein diet, we found that type 3 innate lymphoid cells upregulate IL-22, and this is a negative regulator of the carbohydrate transcriptional program. And during high carb diet, gamma delta T cells actually inhibit the production of IL-22, thereby disinhibiting this carbohydrate transcriptional program. And so to summarize, what we found is that this carbohydrate transcriptional program is induced on demand. It involves cellular remodeling of the intestinal epithelium, and it's regulated by gamma delta T cells, which control this through IL-22. And this work was published earlier this year. And so if you'd like to see more, you can look at our paper that was published in Science last March. And overall, what I think this work shows, especially in conjunction that the work that has been done by many others, is that in response to the environment, be that nutrients or pathogens, the intestine must adjust the balance between its two core functions, nutrient uptake and host defense. And how does it adjust this balance? So we know that in response to environmental pathogens, lymphocytes play a critical role in the intestinal response in increasing barrier defense. And this is through interactions between lymphocytes and the intestinal epithelium that can alter the cellular composition of the epithelium to increase the frequency of barrier and host defense related cells. And what we found is that in addition to regulating the intestinal response to pathogens, lymphocytes also regulate the other major function of the intestine, which is nutrient uptake, by interacting with the epithelial cells to change the cellular composition of the epithelium and ultimately allow this tissue to adjust the balance between its two major functions, nutrient uptake and host defense, in response to environmental change. And so to conclude, I just want to come back to this pair of videos and to remind you that digestive systems constantly adapt to environmental change. And I think that the work that we've done, as well as the work of many others, shows that the involvement of immune cells is really critical for the ability of the intestine to adapt to different environments. And with that, I'd like to acknowledge my mentor, Ruslan Medzatov, and people in the Medzatov lab that helped me with this project, as well as my current postdoc mentor, Catherine Dulac, and the folks in the lab here that I work with, my funding from HHMI for my postdoc and the NSF for my PhD. And if anybody would like to contact me, this is where you can find me online. And with that, I will take any questions.
Thank you, Dr. Sullivan. What a great talk on the dynamic regulation and nutrient handling, with a fascinating role, of course, for the immune system. Again, it rules everything, right? And we do have some questions for the audience. I think the effect of diet on the gamma delta cells will have a downstream effect on the response to pathogens in the gut.
Yeah, this is a great question, and there are a few current postdocs in the Medzatov lab that are studying this exact question. So, as many of you probably know, IL-22 is a really important antimicrobial cytokine, so it plays key roles in protecting against bacterial infection. And of course, we see that the levels of IL-22 can be regulated by diet. And so it's interesting to speculate that perhaps there might be an interaction between diet, specifically high protein diet, and antibacterial immunity that operates through IL-22.
And kind of similar, Donald Palmer wants to know how much would the gut flora and then how much would age impact some of these transcriptional changes?
Yeah, this is another great question. So we actually specifically tested the role of the gut flora, which I didn't have a chance to get into, but we see that this is actually independent of the microbiome. And so we can perform these experiments in germ-free mice and we see the same phenotype. With respect to age, we haven't looked at that in a ton of detail. What I can tell you is that the experiments were done in mice that are between like 7 and 9 weeks of age. And when we look at younger mice, we don't see as strong of a regulation by diet. So this suggests that maybe the gut needs to be mature, the animals need to be fully adult in order to see these sorts of dynamic programs. But how does it change with aging specifically and much older mice? It's not something that we've tested, but it's a really interesting idea.
Thanks. Neil Silverman is asking, what about the TCR on these cells? Any chance they're directly sensing? Or I kind of thought, is it an immunometabolism thing? Is this a bioenergetic standpoint? Do you have any idea?
Yeah, so we, in our single cell sequencing data, we actually did see a strong skewing of the TCR towards V-gamma-6. And so, we don't know much about the specific antigens that are detected by different types of gamma delta TCRs. And this is, a really active area of research in the gamma delta field. And we've thought about collaborating with others to sort of that specialize on this type of work to answer these questions. But it does suggest that there's a role for the TCR in this phenotype. And it'll be interesting to see, with folks that specialize in TCR antigen interactions to understand whether the TCRs are directly sensing diet. And about the metabolism question, our collaborator, Daniel Musita's lab, had a paper several years ago showing that the metabolism specifically of gamma delta T cells can alter the production of different cytokines. So we think that probably the metabolism of the gamma deltas may also be playing a role here, but it's not something that we directly tested ourselves.
Have a bunch more. I'll ask one more question and then we'll go on to the next session, get everyone ready for that. Juliet Morrison wants to know, do you think that targeting the immune response in the gut could be useful for weight loss since nutrient uptake is then dependent upon those immune cells?
Yeah, this is also a really interesting idea, especially given the sort of IL-22 connection. So in a 2014 or 2015 paper, they found that just treating animals with IL-22 during diet-induced obesity could protect the mice from insulin resistance and other effects. And so it's really tempting to speculate that if we, get rid of gamma delta T cells and thereby, change the levels of IL-22, this might alter weight gain in animals just by changing directly nutrient uptake in the epithelium. It's not something that we've tested, but I think especially the changes that we see in whole body metabolism suggest that this could be, a viable strategy, at least in mice. Humans are obviously another story. We don't want to go around doing immunotherapy in people if we don't have to. But I do think that it's likely that this could be an interesting model for diet-induced obesity in mice.
Thank you so much. And thank you to all of our speakers for sharing their work today, and for all of the attendees. We hope you'll stick around for the DEI Initiatives panel on next. And I know everyone's preparing in the background for that. So see you all in a few minutes. Thank you.